Welcome everyone, Mike Fave here. Today I'm going to go through an interesting article that was actually something that I looked into for one of my clients, and it discusses the mechanism of different plant compounds, specifically polyphenols, on protein metabolism or putrefaction by bacteria or microbes in the intestine. The title we have here today is Interference of Dietary Polyphenols with Potentially Toxic Amino Acid Metabolites Derived from the Colonic Microbiota. So basically, I'm going to get into it here. I'm going to try to keep this to about 15 minutes or so. So we're going to start with the general idea. So they start here: each day, varying amounts of undigested or partially digested proteins, such as the colon, reach the colon, where they are metabolized by microbiota, resulting in the formation of compounds such as ammonia, p-cresol, scatol, phenol, indole, and hydrogen sulfide. So these are the toxic compounds that are going to be covered in this paper. So essentially ammonia, p-cresol, scatol, phenol, indole, and hydrogen sulfide. These compounds are produced by the breakdown, as they discussed here, of proteins reaching the colon and the microbiota basically getting a hold of the proteins and amino acids, and then they're through their metabolism and converting them to these compounds. This topic is actually extremely important for people who are on carnivore diets, people who are on low-carb diets, keto diets, who have any type of IBS or digestive disorders, ulcerative colitis, Crohn's, or are like athletes on high-protein diets, because the high protein intakes of a lot of these diets, or the dysbiosis that some of these people have in these particular situations, lead to excessive production of these compounds. And as we're going to get to in the paper, these compounds can damage the intestine in multiple ways and have negative effects on other areas of the body as well. So it's important to figure out how do we modulate these compounds, because obviously there's a threshold for protein that we want to have, but there are negative effects that come with the protein reaching the colon, and with more protein in the diet, you're going to have a byproduct of more protein in the colon. So how can we minimize that? What they go into here further, they say in humans, their accumulation of these products is potentially harmful, and has proposed that they can be involved in the development of pathology such as colorectal cancer and ulcerative colitis, among others. This review assesses the evidence supporting the use of dietary polyphenols to reduce the production of these metabolites. Most studies have used condensed, which is the proanthocyanidins or hydrolyzable, so the allagitannins and gallotannins, and have been carried out on farm animals. Several show the administration of tannins in pigs, chicken, and ruminants decrease the levels of ammonia, p-cresol, scatol, and/or hydrogen sulfide, improving meat and milk quality and reducing manure odor. Few studies were carried out in rats and humans, and the results confirmed to a lesser extent those reported in farm animals. These effects would be due to the capacity of tannins to trap ammonia and hydrogen sulfide and to modify the composition of the microbiota, reducing the bacterial populations producing metabolites. In addition, the proanthocyanidins, so the PACs here is proanthocyanidins, they're a type of polyphenol or a type of tannin, prevent p-cresol and hydrogen sulfide-induced alterations on intestinal cells in vitro. So essentially, the proanthocyanidins and hydrolyzable tannins, which is the allagitannins and gallotannins, they're beneficial plant compounds that are capable of altering the microbiome to reduce the production of these compounds, namely ammonia, p-cresol, scatol, and hydrogen sulfide. And then they also, more specifically, the tannins can trap ammonia and hydrogen sulfide, and the proanthocyanidins, very specifically, can prevent the formation or can prevent the damage on the intestinal cells of p-cresol and hydrogen sulfide, at least in vitro, which means in a cell dish. But that means that there's a possibility that it could also happen in vivo, which would be in a living animal or human. So what they go into here is a brief overview of the polyphenols. I'm not going to read everything. I'm just going to get into some of the highlighted sections as usual. What they start here, basic overview. The importance of polyphenols in human health is supported by several preclinical and epidemiological studies showing the association between high dietary polyphenol intake and lower risk of metabolic, cardiovascular, tumoral, and neurodegenerative diseases. The most abundant flavonoids are tannins, which are present in plants, bark, wood leaves, seeds, fruit skins, as well as in numerous plant-derived foodstuffs such as red wine, green tea, cocoa, cranberry juice. Grape juice is another one instead of just red wine. Pomegranate juice is a great source. Berries are great sources. A lot of vegetables are great sources. So basically, fruits and vegetables and spices are excellent sources of these plant-derived polyphenols, flavonoids, or tannins. A lot of them are actually in the same categorization as each other. So they're saying these tannins are the fourth most common group of compounds in vascular plants, where they constitute up to 20% of dry weight. So they're in the plant foods, they can be quite high. There also are some polyphenols that get deposited, or some plant compounds that reach animal products through their diet, but hands down the highest amount that you're going to get is from the plant-based foods, and that's going to be your vegetables, your fruits, and your spices. What they describe here is, in humans, intakes of 54 milligrams per day and 450 milligrams per day have been reported in the USA and Spain, respectively. In animals, a study in the European roe deer showed that feeds ingested by these animals contained about 1.4% dry weight of condensed tannins, representing an approximative intake of 6 grams per day with high variability according to the season of the year. So essentially, in the animals in the wild are eating significantly more of these tannins than we are. Obviously, for a deer, it's going to be higher because they're eating all plant foods. Now, the interesting piece here is, for humans, Americans have about 54 milligrams per day, and the Spanish have about 454 milligrams per day. So almost 10 times as much polyphenol intake in the Mediterranean as in the USA. And there's all this talk about the Mediterranean diet, the whole grains and this and that, whatever it is. I would guess that a large benefit of the Mediterranean diet would be from this increase of polyphenolic and tannin and plant compound intake, and over the American diet, which is like negligible amount. This is probably from the cup of coffee that somebody drinks in the morning. And that's basically all the plant compounds they're getting in the diet the rest of the day, or in the standard American diet, because they're mostly eating processed foods, which remove a large portion of these types of compounds. So I would say there's a lot of things that people hypothesize as the benefit for Mediterranean diet. I think a large portion is some of these plant compounds and a lack of some of the crap that Americans get. But I think that there's better diets in the Mediterranean diet, and there's factors that can be modulated in the Mediterranean diet to make the Mediterranean diet better in and of itself. But I'm not going to get into that now, so that I don't spend like 2 hours going through this video. So we're going to continue here. The next thing they say is most polyphenols. So this is actually an extremely important point. Polyphenols are not absorbed in the small intestine. Hands down, by far, most of them are not even close to absorbed. Most of them reach the colon, where they're fermented into different compounds, and then they modulate the microbiome in beneficial ways. So the benefit is largely through the modulation of the microbiome, and then perhaps some of the compounds that are created by the microbiome, not necessarily the initial compounds and us absorbing them. So they're basically, I just did an Instagram post on this, but they're microbiome curators, which is extremely important. So the medicinal effects of them are largely through those mechanisms. There are other mechanisms when they are absorbed, but that's beyond the scope of this this article. So they go on to say most polyphenols are poorly absorbed in the small intestine and reach the colon, where they are metabolized by the microbiota. It is considered that proanthocyanidins with a degree of polymerization greater than 2 are too large to be absorbed and accumulate in the intestinal lumen. This is supported by a study reporting that 90% of proanthocyanidins from apple juice ingested by ileostomized patients were recovered in their ileostomy effluent. In the colon, glycosylated polyphenols are hydrolyzed, releasing diholosides and oligosides, which are rapidly fermented by the commensal bacteria, resulting in the formation of short-chain fatty acids. So, couple things here. The proanthocyanidins, polymerization is basically the linking of two groups. So a monomer would be one element, a polymer would multiple elements. So if you have greater proanthocyanidins with a polymerization greater than 2, they can't be absorbed by our small intestine. The molecule is too large to be absorbed. And what they basically found was in people who have ileostomy. So an ileostomy is essentially where you take the ileus, which is part of the intestine, and you cut it and you bring it to the abdominal wall. And so stool comes out through your abdominal wall into a bag rather than going into a colon. Now, they didn't give this to people for the study. Obviously, they had people who had ileostomies. Maybe they had a colon resection. Maybe they had some type of colon rupture, and they needed to get it to repair, whatever it is. When there's issue with the colon, they usually create an ileostomy. Maybe they had some type of wound and that they didn't want them to produce bowel to produce stool anymore, to get into the wound on their sacrum or whatever it is, so they have an ileostomy. There's multiple reasons why, but essentially what happens is you basically get the contents of food that are leaving the small intestine. They never make it into large intestine. They go into a bag, so you can see what's left over after people's bodies have tried to absorb all of the nutrients. And what they're basically showing is that 90% of these proanthocyanidins, these tannins, polyphenols, aren't absorbed in the small intestine. So what they go on to say is that in the colon, the polyphenols are hydrolyzed, so they're broken down, and then they release these diholosides and oligosides, so these different compounds that the bacteria break them down in the colon because they have enzymes that can break them down. We don't. And then they basically can ferment the breakdown products to form short-chain fatty acids. Now, short-chain fatty acids are extremely important because they serve as the energy substrate for the colonic epithelium. And then they also help to acidify the colonic lumen, which helps to prevent the formation of pathogenic bacteria taking up residency inside the colon. So they're actually, to a degree, prebiotic. They're antimicrobial, they're bacteriostatic, and then they're also prebiotic, and we'll get into that in a second. What they finish up saying here is, while the concentration of polyphenols in fecal water from healthy volunteers are around 2.7 micromoles with high inter-individual variability, the total concentration of flavonoid-derived microbial metabolites is more than 200 times. The complete metabolism of phytochemicals requires, hence, specific microbial consortia that intervene according to the enzymes they express. So essentially what they're saying here is that in fecal water, there's not really a high concentration of polyphenols. The reason why is because the bacteria express a whole bunch of enzymes that can break down these polyphenols and create a whole host of other compounds. And essentially, specific bacteria are able to use specific compounds. So by bringing in a large intake of polyphenols, you're basically curating your microbiome. You're allowing for the growth of certain species of bacteria and not others, and perhaps destroying or eliminating other pathogenic species. It's like if you have a zoo and you want a whole bunch of monkeys, you might as well throw in bananas. If you wanted to have a whole bunch of lions and you want to feed it meat. If you don't want lions, then you wouldn't if you wanted lions in there, you wouldn't throw them bananas. It wouldn't feed the lions. They wouldn't be able to thrive. So it's similar. It's a very crude analogy, but similar type of idea inside the intestine. So what they further on go and say bioreactor and animal studies indicate that in the presence of polyphenols, the relative abundances of beneficial bacterial populations involved in these metabolic processes increase, while other potentially harmful populations, pathobionts, decrease. These results have been confirmed by various clinical trials showing that dietary supplementation with polyphenols from green or black tea, coffee, berries, cocoa, red wine, apples, cereals, or soy stimulates the growth of bifidobacterium, lactobacillus, and eventually that of streptococcus, bacteroids, and bacteria-producing butyrate such as Faecalibacterium prausnitzii and Eubacterium rectale, recognized for their anti-inflammatory effects and their ability to regulate energy metabolism. So I'm going to stop here and I'll read the rest after. Basically, the polyphenols in these substances, green or black tea, coffee, berries, cocoa, red wine, apples, cereals, or soy, stimulate the growth of these are all beneficial bacteria that have different, that produce different compounds that have a health effect inside the colon as well as throughout the rest of the body. Even the health effect could be destroying another bacteria, crowding out another bacteria, or there could be specific compounds that can be created that the body can use. Now, I would give caution to soy specifically because there are some studies showing that soybeans actually cause dysbiosis and overgrowth of E. coli inside I'm pretty sure it was soybeans, inside the small intestine. So I would be very careful with soy. If you're going to use any soy, it should be fermented soy products, if any at all. And some of the polyphenols or the plant compounds in here, the isoflavones, they're phytoestrogens, so they're not as potent as estrogen, but they're also goitrogens, so they can minimize thyroid function, so not great overall. And then cereals, even though they have polyphenolic compounds, they also have other fibers and compounds like lectins and phytic acid, etc., that can be damaging for the intestine. So if you want to focus on they're not as high as these other foods. Same thing with soy. So if you wanted to get higher amounts of polyphenolic compounds, or these plant tannins, I would go with the teas, coffee, berries, cocoa, red wine, apples, other fruits besides apples. Doesn't have to be apples. Different spices are extremely high in polyphenolic compounds. So those are just some basic things. And also vegetables are as well. So just things to keep in mind. And there's multiple different polyphenols. There's multiple different tannins. There's different types. They have different effects on the intestine. So just things to keep in mind. I'll continue from here. So they say, in addition, some of these studies report a decreasing abundance of potentially pathogenic populations of C. histolyticum, so Clostridium histolyticum, Clostridium perfringens, Pseudomonas, and Enterobacteria. So these are all pathogenic species that can cause disease as well as different issues inside the intestine. They go on to say, such decrease could be due to direct bacteriostatic or bacteriocide activities against commensal bacteria and food pathogens. For example, proanthocyanidins were shown to inhibit the growth of Enterobacteria, E. coli, and Salmonella, maybe through their ability to bind lipopolysaccharide, and gram-positive bacteria including Enterococcus, Staphylococcus, Streptococcus, Klebsiella pneumoniae, Pseudomonas aeruginosa, Clostridium perfringens. So all of these are actually like known pathogens, not only in the intestine but in other areas of the body. They're on wounds. They cause spoiling of food. They can be causing pneumonia. They can be causing a whole host of issues. So these polyphenols are actually able to inhibit their growth, and they can bind lipopolysaccharides. So they can directly bind endotoxin, which is actually quite helpful. So they can eliminate these pathogenic bacteria, the polyphenols, and then they can also stimulate the growth of these beneficial bacteria, which is a multifold effect because the beneficial bacteria can then further crowd out these pathogenic bacteria while the polyphenols are also inhibiting their growth altogether. Plus, the compounds that can be produced by the metabolism of the polyphenols from these bacteria may have beneficial effects like the short-chain fatty acids on the colon. So polyphenols have a multifold prebiotic like ecosystem curating effect, more so than just this like random fiber like xanthan gum, which can encourage the growth of multiple different species of bacteria, possibly some pathogens. So that's this selective process is extremely helpful. The other thing that's interesting is that the polyphenols are able to improve antibiotic resistance in the sense that when you add certain polyphenolic compounds or essential oils or some of these other plant compounds with antibiotics, they increase the effectiveness and they block antibiotic resistance. So altogether, there's definitely they definitely have beneficial effects overall in specifically modulating the gut. So now they're going to get into proteins in the GI tract, kind of setting the stage for how these other compounds that we're going to get into are created. They start here, they say the small intestine is highly efficient in digesting dietary proteins, and it is generally considered that 90 to 95% of them are absorbed into the bloodstream. The digestibility of proteins is, however, variable depending on their origin, animal or vegetal, the food matrix where they are included, and the time and type of cooking of protein-containing foodstuffs. It must be considered that 20 to 40 grams of endogenous proteins issued from salivary, gastric, and biliopancreatic secretions, enterocyte desquamation, secretions from the systemic compartment, and bacterial recycling also transit through the digestive tube. Globally, it is considered that around 10 to 12 grams of undigested or partially digested protein reach the colon daily, where they are metabolized by commensal microbiota, being these amounts higher in individuals consuming higher protein diets. So the food that you eat, the protein that you eat, a portion of that will reach the colon, and then a portion of the proteins that are just inherent in your digestive tract from your digestive processes, including salivary secretions, gastric secretions, secretions from your pancreas and from your gallbladder, loss of the top layer of the intestinal lining, secretions from systemic compartments, so from the just like the rest of the organs, etc. Bacterial products, all those there's all proteins in those areas that can reach the colon beyond food. Interesting note: if you don't eat, you can still produce stool, and that's from some of these compounds moving through the digestive tract. So they're saying that about 10 to 12 grams of undigested or partially digested protein reaches the colon daily. Obviously, if you increase your protein intake, you'll have more. The little diagram here is describing some of these different elements. So essentially, what they're saying here is that the tannins can bind some of the some of the proteins in the digestive tract, and they can decrease their absorption because they form like these insoluble complexes with the protein, and they can also, some of the tannins can also inhibit digestive enzyme activity. So this is not necessarily ideal, but these effects over here are pretty ideal overall. So while tannins can decrease protein absorption a bit, they can also inhibit the bacterial utilization of the proteins in the colon, specifically the harmful bacteria, and then they can increase the beneficial bacteria and also decrease the harmful bacteria. So they have this modulating effect. So all in all, you're still going to absorb the protein with the tannins and polyphenols. You may have a slight decrease in absorption, but their curating effect on the microbiome is important, decreasing lipopolysaccharide, decreasing pathogens, increasing beneficial bacteria, and then decreasing some of these toxic compounds we're going to get into now. So they are going to lay the stage here now for some of the amino acid metabolites. So they start here. In addition, amino acid fermenting bacteria can produce ammonia, polyamines, phenol, indole, scatol, P-cresol, and hydrogen sulfide, most of them being potentially harmful to the colonic epithelium, depending on their concentrations. These metabolites are rapidly absorbed, and for some of them, sulfated or glucuronidated in the colon and or liver and excreted in the urine. Their urinary excretion is highly variable in healthy adults, ranging from 10 and 55 milligrams for P-cresol and 4 and 7.5 milligrams per day for phenol, and are higher in obese subjects. Noteworthy, indoxyl sulfate and P-cresol sulfate are considered uremic toxins, which accumulate in the bloodstream of patients with chronic kidney diseases, contributing to the aggravation of renal damage and development of cardiovascular complications. So, bacteria can basically take the amino acids and create these compounds. These compounds are absorbed. Some of them are absorbed and they're basically detoxified. So a sulfate or a glucuronic acid group is added to them, either at the colonic epithelial cell or at the liver, and then they're excreted. Now, some of them are excreted by the kidney, and they are toxic to the kidney, such as P-cresol sulfate and endoxyl sulfate. So once they're sulfated, so that they can be made polar and then water soluble to be excreted in the urine, they're still toxic to the kidney. When your kidneys don't work, such as if you're on dialysis, end-stage renal disease, you have an acute kidney injury, etc., these compounds can build up because the kidney is not filtering them, and if it is filtering them, it's kind of damaging the kidney, thus decreasing function, and then they build up in the bloodstream, and then they can also damage the vasculature, leading to those cardiovascular complications, as well as the negative effects on the heart of the kidney not functioning appropriately and filtering out the blood. So, first compound we're going to get into, ammonia. Intraluminal colonic ammonia originates from amino acid catabolism, protein synthesis, and urea degradation by bacterial urease. Gram-negative bacteria are considered the main contributors to ammonia production. So gram-negative are the ones that produce endotoxin. Gram-positive produce other toxic compounds like lipotecoic acid, but the gram-negative ones are the ubiquitous endotoxin creators. A note on endotoxin, there's different types of endotoxin with different structure that trigger the immune system in different ways. So all endotoxin isn't great, but there's a spectrum of toxicity of different endotoxins. A lot of people just think endotoxin is one thing. It is, in general, one thing, like one category of thing with different subtypes inside the category based on the bacteria, just like bacteria are one category with different types inside that category as well. So what they say is high colonic levels of ammonia were shown to inhibit the expression of monocarboxylate transporter 1 implicated in butyrate absorption. So basically, high levels of ammonia inhibit the epithelial cells in the colon from absorbing butyrate. This is a problem. Why? Butyrate is one of the main substrates for the colon to use as an energy source. They go on to say, this finding could be highly relevant considering the importance of short-chain fatty acid in the regulation of colonic homeostasis. In addition, the concentration of ammonia positively correlated with mucosal interleukin 8 and tumor necrosis factor alpha, suggestive of its associations with mucosal inflammation. Similar results were also observed in a study with the polyamine putrescine. Reinforcing these results, ammonia was also shown to decrease butyrate oxidation in colonocytes, reducing ATP production and increasing glycolysis, finally resulting in alterations of the barrier function. Excessive production of ammonia also contributes to the development of hepatic encephalopathy. So a couple things here. The ammonia and then a polyamine, which we'll get to in a sec, called putrescine, increased inflammatory markers of the mucosa of the intestine. Specifically, these are cytokines, tumor necrosis factor alpha, and interleukin 8. These you can see in like infective processes to some extent as well. And then they also, specifically ammonia, basically decrease the colonocytes' ability to produce energy. And then when the colonocytes are unable to produce adequate energy, their ability to maintain a barrier in the intestine fails. So when bacteria are producing ammonia and some of these other toxic protein compounds, what winds up happening is it can cause barrier dysfunction in the colon and let these substances go across the barrier because it impairs their energy production. And then essentially all blood supply goes to the liver from the intestine and the spleen. So the liver basically bears the brunt of this and is forced to detoxify these different compounds. And that's why you see in alcoholic liver disease or cirrhosis or any disease that kind of heavily impairs the liver, you can see something develop called hepatic encephalopathy, which is essentially loss of mental function because the ammonia level in the blood gets too high and because the liver can't clear that ammonia. So the liver is usually clearing this ammonia. Something else that could be more relevant to people who aren't extremely sick with liver problems, if you have any types of gut issues or dysbiosis or anything like that, you'll notice that you may have blood sugar regulation issues, mood issues. That could be a sign of impaired liver function because the liver is supplying that blood glucose through its glycogen for the brain. So if you have stuff going on in the gut, you have all this inflammation, these compounds being created, the liver is getting the blood from the gut, and then it's like fixing what's going on, and then that can impair your mood, that can impair your blood sugar regulation, that can impair hormonal regulation, it can impair detoxification, etc. So you really want to make sure like the gut and the liver are intimately related. So if you're having all these weird mood, blood sugar, sleep, insomnia, all these types of situations, it could be something going on in the gut that's causing an issue with the liver or something more specifically with the liver. But usually they go hand in hand. If the liver's not working well, there's issues with the gut. If the gut's not working well, the liver gets beat up a bit. So we'll jump forward here. We're going to talk about indole and scatol. So indole is formed from dietary tryptophan by the action of tryptophanase, an enzyme expressed in many bacterial species, including bacteroids, Clostridium, and E. coli, Escherichia coli. So indole, there's not so much to talk about. They discuss it as having some beneficial effects, but scatol is really the bigger issue. They say it's a malodorous metabolite that originates in the colon from the decarboxylation of indole acetic acid by bacteroids and Clostridium species. In humans, the fecal concentration of scatol ranges from 0 to 5 to 80 to 100 micrograms per gram, according to the intake of animal protein and particularly of red and processed meat. So red meat can increase scatol. When present in high levels, scatol can act as a pulmonary toxin in some species, including humans. On a biological point of view, scatol has been shown to induce apoptosis-associated epithelial cell death, probably by activating the transcription of the aryl hydrocarbon receptor and subsequent increased expression of CYP1A1, a typical aryl hydrocarbon receptor target gene in the colonic epithelium. So essentially, scatol can cause the epithelial cells to undergo apoptosis, and then that can impair barrier function. So just something to keep in mind. I've worked with quite a few carnivores. They've had very high zonulin levels, which is a marker of intestinal permeability. Higher it is, generally the more increased intestinal permeability you have. Now, what they're saying here is when you eat high amounts of red and processed meat and you're not having it with polyphenols and you're not having it with fiber and you're not having it with plant compounds, you can generate large amounts of scatol, and then this large amounts of scatol can cause issues with the epithelial cells inside the colon. So this is very important for keto and carnivore people to understand because the diet is essentially just crap tons of meat and protein and no plant compounds at all, which I think is why a lot of people develop issues on the carnivore diet and increased intestinal permeability, which I've seen with clients as well. Obviously, that's N equals, 20, 30, 40, whatever it is. But yeah, I mean, I can't even tell you how many carnivore people I've worked with over this past time. Most recently, I can't even tell you the number, but that I've seen it pretty consistently with people coming off the carnivore diet and having these the stool test with high amounts of zonulin levels and having dysbiosis, elevated secretory IgA levels. Calprotectin usually isn't high in a lot of these people. That's a marker of intestinal inflammation. And then just seeing like the really weird bacterial profiles that aren't necessarily good. So that's something to keep in mind, like increased hydrogen sulfide producers like disulfovibrio piger. Like that's one of them. So yeah, I think it's between 20 and 40 carnivore people that I've worked with who have gotten stool tests, and I've been able to see it, and I've seen those results. I've worked with more, but not everybody gets stool tests because they're expensive. Somewhere in that ballpark. I'll have to go and look. Anyway, we're going to go on here. This is the next one they talk about is P-cresol. P-cresol is specifically produced from L-tyrosine by anaerobic bacteria, a process that occurs preferentially in the distal colon where protein breakdown is higher. The reason protein breakdown is higher in the distal colon is because in the ascending and transverse portion of the colon, a lot of the soluble fibers and the harder to digest, the rapidly digested fibers can get processed in the ileum and the ascending colon, and then the harder to digest or the more complex fibers take longer to break down, and so those will kind of slowly get broken down throughout the colon in the transverse and some of the descending portion. And then once all those fibers are kind of broken down, the protein usually gets fermented in the distal portion. So one of the strategies to manage issues with problems in the sigmoid colon and distal portion of the colon, and I get to the tangent after, is to basically make sure that you have different types of fiber sources that will be able to make it to those regions of the colon, so you don't have a heavy amount of protein putrefaction without these other fibers and polyphenolic compounds there to protect against the negative effects. So the side note, the tangent is I think a lot of times people develop the diverticulitis in the sigmoid region of the colon because those protein compounds over time can damage the colon. So through these indole, scatol, P-cresol, hydrogen sulfide, ammonia, etc., they constantly cause issue in the colon, and the colon degrades over time. Its structure degrades with the metabolic degradation, etc., and then that can lead to these issues. So one of the ways to solve it is to have adequate amounts of polyphenolic compounds and tannins, and then different fibers that can help reach that distal portion of the colon. So they continue here. They say it's intercolonic. P-cresol's intercolonic concentrations increase in individuals consuming high-protein diets, while they decrease in those consuming diets rich in resistant starch or pre- and probiotics. Exposure of the human colonic epithelial cell line to P-cresol inhibits cell proliferation and respiration, decreases ATP content, and increases DNA damage. Accordingly, urinary P-cresol excretion is higher in patients with colorectal cancer as a reflection of its higher production in the colon. P-cresol can also disturb the mitochondrial function in hepatocytes, suggesting a possible role in the development of hepatic alterations. The sulfate conjugated form of P-cresol is considered uremic toxin involved in the development of renal and cardiovascular complications in patients with chronic kidney disease. So P-cresol can decrease ATP production and cell respiration in colonic cells and increase DNA damage, which would increase the risk of cancer. Then what you're seeing is over time, people with colorectal cancer have a higher amount of P-cresol excreted in their urine, which would mean that there's higher amount formed in the colon because it's not formed anywhere else. And then also P-cresol is a toxin to the kidneys and is implicated in the development of further renal and cardiovascular complications in chronic kidney disease, because in chronic kidney disease, essentially the kidneys don't function so well to remove all these toxins, so they build up in the bloodstream, and then that damages the kidneys, the heart, and the vasculature even more. So yeah, don't get chronic kidney disease. Make sure your kidneys work over time. The microvascular complications of chronic kidney disease I see on a pretty regular basis in the hospital, and they can be pretty bad. Pretty bad diabetes, amputations, blindness, pretty nasty wounds that goes with the amputations, neuropathy, etc. It basically just rots you from the distal portions inward. So the first things to go are feet and hands and fingers, and then you start to lose the lower leg, so it's your shins, and then your thigh, and then you're up at the hip. So yeah, it's the chronic kidney disease and diabetes, they often go hand in hand. They're not a good it's not a good combination. And then obviously there's tons of cardiovascular events that follow that: heart attacks, strokes, et cetera, et cetera, et cetera. Anyway, next thing they go into is polyamines. So there's putrescine, spermidine, and spermine. Some of these are actually beneficial, and they do or can have beneficial effects, and they do discuss this. They say there are small cationic molecules that can be found in the colonic lumen, being putrescine the most abundant, followed by spermine and spermidine. They are produced by different bacteria from collectic biosynthetic pathways involving a complex system of metabolite exchange. These allow the transformation of arginine to putrescine and subsequently spermidine and spermine through the citrulline L-ornithine or the agmatine pathway. So basically they're created from arginine, but there's multiple steps that they have to go through. So arginine is amino acid, in case anyone wasn't aware. Polyamines have been shown to exert multiple physiologic roles in the organism, including DNA and RNA stabilization, protein synthesis, and regulation of mitochondrial function, cell growth, and differentiation. So they can have beneficial effects. They say they also inhibit aberrant gene methylation, alleviate inflammation, and stabilize gut barrier function. However, polyamine metabolism, so the creation or generation of these and utilization of these, can generate toxic byproducts, including hydrogen peroxide, aldehyde, and ultimately acrolein, that affect mitochondrial function and cellular processes and could explain their crucial role in the development of carcinogenesis. In addition, spermidine was recently shown to stimulate the production of colobactin, a genotoxin expressed by some strains of E. coli and associated with DNA damage and tumor promotion. So the presence of spermidine can cause E. coli to produce a toxic compound that can damage DNA and increase tumors, or possibly increase tumors. And then the generation of the polyamines creates other toxic byproducts that are not good, like hydrogen peroxide, aldehydes, and then acrolein. So these products are pretty toxic overall, not ideal to have floating around, even though the polyamines may have some beneficial effects. Last one we're going to get to is hydrogen sulfide. This one there's a lot of this going around in the gut sphere now, is a hydrogen sulfide SIBO. This is actually can cause a lot of issues for people. Basically is like the sulfur farts. So if you're having sulfur farts, it's probably related to the hydrogen sulfide. Obviously scatol has a nasty smell as well, as they discussed, but they go on to say gut hydrogen sulfide is synthesized by epithelial cells and some bacterial populations from several dietary and endogenous sulfur-containing compounds, mainly L-cysteine. So foods high in L-cysteine, if you have an overgrowth of hydrogen sulfide producers, this could be Bilophila wadsworthia, but the most specific ones is the sulfovibrio genus or species, the genus, yeah. And then the sulfovibrio piger is the one that you see on most stool tests. And so if you're having high protein animal products that are high in cysteine, which is a lot of the animal products, can cause issues specifically here. So they say sulfate-reducing bacteria, mainly from the sulfovibrio genus, use hydrogen lactate or formate as electron donors for reducing sulfates. Hydrogen sulfide may also be produced from cysteine or sulfite by the enzymes cysteine desulfohydrase and sulfate reductase, which are expressed by other taxa such as E. coli, Clostridium species, Enterobacter, Staphylococcus, or Fusobacterium, in frequent association with digestive diseases. So hydrogen sulfide is produced by this list of bacteria, so it's actually quite a few. Desulfovibrio is the main one, and then it's associated with digestive diseases. At low micromolar concentrations, hydrogen sulfide acts as an inorganic oxidative substrate for colonocytes. So the colonocytes can produce energy from hydrogen sulfide. At these low concentrations, hydrogen sulfide was shown to stimulate mucus production, restore gut microbiota biofilms, and alleviate inflammation, contributing to the protection of the gut barrier. So in small amounts, it seems to be okay. However, when hydrogen sulfide is produced in excess by gut microbes, it can bind to cytochrome C oxidase complex 4 of the mitochondria, decreasing O2 consumption and ATP production. The clinical relevance of hydrogen sulfide overproduction in colon is related to its possible mutagenicity and implication in the genesis of ulcerative colitis and colorectal cancer. In patients with inflammatory bowel disease, increased hydrogen sulfide production is concomitant with the downregulation of enzymes implicated in mucosal detoxification, while in colorectal cancer patients, upregulation of both endogenous and microbial bacterial amino acid-derived metabolites was reported. So in higher amounts, hydrogen sulfide damages energy production, specifically at cytochrome C oxidase, and then this essentially can lead to mutagenicity, which is cancer or damaging of the DNA, and it's implicated in large amounts of colorectal cancer and ulcerative colitis. So basically, what they're and also in patients who have inflammatory bowel disease, Crohn's disease, ulcerative colitis, when you have higher amounts of hydrogen sulfide, you can see a downregulation of enzymes involved in detoxification, and an upregulation of, well, basically they're saying an increase of these. You see with increased hydrogen sulfide, increased amino acid derived metabolites. These are the metabolites that we've discussed, and a lot of them are toxic, so that's not a good thing overall. So now what what we're going to get into is how these tannins have a beneficial effect on some of these compounds, and we've basically discussed all the compounds, or not all the compounds, but a large amount of the toxic ones that can be created by protein putrefaction in the colon. So now we're going to talk about how to kind of solve this. What are things that we can do? So they start with proanthocyanidins have been shown to bind bacterial lipopolysaccharide in vitro and reduce the postprandial absorption of these pro-inflammatory compounds. So the solution here, as the paper started in the beginning, is these polyphenols, these tannins. And so we're going to discuss the mechanisms by which they have the benefit for these protein compounds. So first off, proanthocyanidins can bind endotoxin and make it less absorbed. Furthermore, they say tannins can also react with other small molecules containing two or more, I think it's amino residues, such as polyamines, putrescine, spermine, and spermidine, and with the nucleotide bases and amino sugars, chitin and chitosan. So the tannins kind of bind possibly negative things inside the intestine. So they go to say, therefore, tannins would act as trapping mechanisms for ammonia that could explain the reduction of ammonia emissions by proanthocyanidins described at continuation. So the proanthocyanidins can bind lipopolysaccharide and they can bind ammonia and basically make it not absorbed, which is awesome. So this is things you could find: proanthocyanidins, grapes, blueberries, elderberry, teas, cranberries. I think all the pomegranates, I think. So having grape juice with a meal, pomegranates, some blueberries, these different foods can be quite helpful to bind up some of these toxic compounds. So if you're going to have a fat steak, you're going to have a lot of protein in one sitting. Maybe have some grape juice and some blueberries. Grape juice on the side, blueberries for dessert. Maybe some chocolate as well with your blueberries, and that can help to minimize some of these toxic products. What they further say is, respective to hydrogen sulfide, Olson et al. Have observed that blueberry, bilberry, and cranberry polyphenolic extracts, as well as pure polyphenols, rosmarinic acid, cyanidin, quercetin, and resveratrol, are auto-oxidized to semiquinones or quinones that subsequently oxidize hydrogen sulfide to polysulfides and thiosulfate. The resulting molecules are potent free radical scavengers that can initiate a downstream antioxidant response and cytoprotective effect through KEAP1 persulfidation and NRF2 activation. Similar results were reported with T catechins. So the polyphenols from blueberries, bilberries, cranberries, as well as pure polyphenols, so rosmarinic acid, cyanidin, quercetin, and resveratrol. Resveratrol is grapes, quercetin's onions, cyanidin, I don't know which one it is, rosmarinic acids. And they're all in quite a few, but these are the main ones, is rosemary, but there's also other foods that it's high in. So they interact with the hydrogen sulfide and create polysulfides and thiosulfates that are free radical scavengers, and then they also upregulate the detoxification pathways, or technically hermetic pathways of the cells, to some extent. So, and which does show antioxidant effect and a cytoprotective effect. So these polyphenols can basically help to detoxify hydrogen sulfide specifically. They bind ammonia and they bind lipopolysaccharide. So they're quite beneficial. So they're binding these toxic compounds, killing the negative bacteria, and increasing the growth of the positive bacteria. So they talk about livestock studies here. And then I think they go on to talk about laboratory animals and then humans. In the livestock studies, say dietary supplementation of pigs with a green tea polyphenols extract containing 90% flavonthiols for 2 weeks displayed a remarkable effect in colonic metabolism, preventing the production of ammonia, p-cresol, phenol, and scatol, and enhancing of short-chain fatty acid and lactate, being these changes accompanied by strong attenuation of stool odor. These changes were also associated with higher levels of lactobacillus, lower levels of Bacteroideaceae and the pathobiont Clostridium perfringens in the supplemented animals. So they supplemented them with green tea polyphenols using green tea extract, and it decreased the production of ammonia, p-cresol, phenol, scatol, and increased short-chain fatty acids and lactate, which are fuel sources for the intestine. So they decreased the toxins and increased the fuel sources for the intestine. This is in pigs who have an extremely similar digestive tract to humans. So this is actually relevant to humans to some extent, more so I think than mice. And then basically it increased lactobacillus, which is supposedly a commensal bacteria, and then decreased Clostridium perfringens, which is a known bacterial pathogen. So that's actually beneficial. In another study, the culture of swine fecal bacteria with 0.16 purple sweet potatoes, polyphenols containing 79% of total polyphenols as gallic acid equivalents, mainly anthocyanins, and fibers led to a drastic reduction of p-cresol production without affecting that of ammonia compared with the control culture without polyphenols. This result indicates, therefore, the polyphenols other than tannins may interfere with the production of amino-derived bacterial metabolites. So you have all other polyphenols have an effect. That's not surprising besides just the tannin class. This one was purple sweet potatoes, which basically decreased p-cresol, which we talked about was a kidney toxin and a cardiovascular toxin. It has to be detoxified by the liver or the colonic epithelium and damages the colonic epithelium. So there you go. Now we have purple sweet potatoes on the list as well. This one's in chickens, and they use pomegranate. Basically what they showed is that it decreased ammonia, methanethiol, and to a lesser extent hydrogen sulfide. The supplemented animals had decreased E. coli and salmonella and increased counts of Bacillus C. cerevisiae, which is actually a yeast. And then so basically it increased species of bacteria in the intestine that can express tannases, which are enzymes that break down tannins. And so the pomegranate increased these things, and so increased these species, decreased these toxic species, decreased the toxic compounds. And they say it is therefore probable that pomegranate hydrolyzable tannins could decrease the growth of ammonia-producing and sulfate-reducing bacteria and/or inhibit bacterial urease activity. So there you go. Your steak and your pomegranate juice should be a good combination, or chicken and grape juice, whatever works for you. I'd go with lean chicken to minimize PUFA. Now they're going to talk about rats. They say in another study, rats were supplemented for 8 weeks with 2 raspberry extracts. Compared to control animals, lower concentrations of ammonia were detected in the cecal digesta of the animals supplemented with the extracts containing higher elagitannins, flavin-3-ol ratios, independently of its dosage. In another study in rats, the administration of epigallocatechin gallate, the main phenolic compound present in green tea, for 4 weeks, increased fecal losses of carbohydrates and proteins due to the reduction of apparent digestibility of these macronutrients. We talked about that. But then they also say the cecal concentrations of p-cresol decreased, while that of phenol was enhanced, possibly due to the microbial catabolism of EGCG. These events were accompanied by a strong diminution in the abundance of Clostridium, affecting more particularly the clusters, 11, 14, and 18. So basically, green tea extract in rats decreased ammonia. It did decrease protein and carb digestion a little bit. And then it decreased some of the possibly pathogenic Clostridium species while decreasing p-cresol. So again, a lot of the polyphenols have beneficial effects. We're going to get into humans really quick, and then we'll wrap things up. In humans, they say Yamakoshi et al. Administered their grapeseed extract to healthy individuals and elderly inpatients for 2 weeks. So they're in a hospital. A marked decrease in fecal odor, methylmercaptan, and hydrogen sulfide concentrations was observed compared with the administration of green tea extract and champignon extract. They also reported higher counts of bifidobacterium, where those of Enterobacteriaceae, and the level of ammonia, phenol, p-cresol, 4-ethylphenol, indole, and scatol tended to decrease at the end of the treatment period. So it looks like green tea extract has a beneficial effect, grapeseed extract has a beneficial effect in humans, decreasing these toxic compounds and then increasing bifidobacterium. So that's great. Another study reported that daily intake of 50 grams of dates containing 750 milligrams of polyphenol for 3 weeks decreased fecal ammonia excretion and genotoxicity of fecal water in healthy adults. In the last study, the metabolic effects of 4-day administration of a mix of red wine and grape juice extract were determined in a crossover study carried out in healthy adults. In urine, the main polyphenolic metabolites detected after treatment were syringic acid, 3-hydroxypuric acid, pyrogallol, hydroxyphenolacetic acid, and 3-hydroxyphenylpropionic acid. The urinary excretion of p-cresol sulfate and 3-indoxyl sulfuric acid decreased, while that of indole 3-lactic acid and nicotinic acid increased compared with the control period. So, first of all, just to point this out, 50 grams of dates a day had 750 milligrams of polyphenols. If we come up here to the average American diet compared to Spanish, Americans are having 50 milligrams per day of polyphenol intake. So, yeah, just something interesting to think about. If Americans just had 50 grams of dates a day, or maybe some fruit at all, their polyphenol intake would be significantly higher. And basically what they're showing here is that the date intake or the date intake decreased ammonia and then the genotoxicity of fecal water. So they basically take the stool and they determine if it causes issues to the genes inside cells. And then the last one is the red wine and grape juice extract basically decreased some of the p-cresol sulfate and then indoxyl sulfuric acid. And then so indole 3-lactic acid and nicotinic acid increased. So it changed the metabolism of some of the indoles, and then it decreased p-cresol. So it decreased some of the toxic compounds. So overall, what we're seeing is that the polyphenols decrease toxic compounds, increase beneficial bacteria. They also have other beneficial effects outside of that, which was for other studies. And then we have like some graphics here to kind of break this down. So in rats, indole was increased with some of these beneficial compounds. Hydrogen sulfide, ammonia, and p-cresol decreased. Beneficial bacteria, bacteroids, and lactobacillus increased, while Prevotella and Clostridium decreased. I'm pretty sure Prevotella is a gram-negative endotoxin producer. In humans, what they're basically found is that in fecal cultures, hydrogen sulfide decrease. I'm not sure what this compound is here, unfortunately. They're saying that ammonia, hydrogen sulfide decrease, propionate and isovalerate decrease. So this is a branched chain fatty acid. This is a short chain fatty acid. But butyrate, which is another short chain fatty acid, increase, and also indole, which, as we discussed above, isn't necessarily negative. And then composition of the fecal microbiota. So actinobacteria, Ruminococcaceae, and Fecalobacterium increased. So Fecalobacterium I know is beneficial. Actinobacteria I know has some beneficial effects, not necessarily pathogen. Not sure on Ruminococcaceae. And then this in the in vitro studies, basically the tannins are showing increased mitochondrial function, and then increased cell viability, decreased epithelial permeability, and then they're saying detoxification down, but I don't think that they mean that it's decreasing the cell's ability to detoxify. So yeah, basically the conclusion here, the biggest thing is the beneficial effects of tannin administration are probably related to their capacity to trap ammonia and hydrogen sulfide and act as a candidate prebiotic, modulating the composition and functionality of the gut microbiota. So essentially, these different foods that most people know are good to a large extent or are generally considered healthy, although grape juice necessarily isn't, but the juices are indeed healthy. They do have beneficial effect, have polyphenols and tannins, and these compounds are able to basically bind up the toxic compounds from protein metabolism inside the colon, and also decrease the pathogenic species of bacteria that live in the colon while increasing the beneficial species. So essentially, they wind up being these microbiome curators to a large extent. And so for anyone with a high protein diet, anyone with a carnivore diet, a keto diet, etc., I guess on a carnivore diet, these compounds couldn't be added. But if you care about microbiome health, which is extremely important overall, just knowing the effects of lipopolysaccharide and then the effects of intestinal permeability, incorporating these compounds, these fruits and vegetables and spices in your diet can help to modulate the microbiome in these beneficial ways. As we can see here in this graphic, decrease ammonia, phenol, indole, scatol, p-cresol. We know it decreases hydrogen sulfide. decrease gut protein fermentation, decrease microbial amino acid metabolism. Then it will increase beneficial bacterial species and increase the health of the colonic environment overall. So adding these foods, again, pomegranate, cranberry, grape juice, blueberries, bilberries, elderberries, green tea, black tea, chocolate, all of these different foods can have a beneficial effect. Even they list dates here. I'm sure it's across all of the fruit and vegetable spectrum have these effects. I know for a fact they do. There are other studies discussing them. Maybe they don't all affect hydrogen sulfide or ammonia, but they have other effects on the microbiome in general. So incorporate these foods with your diet. If you're having a protein meal, have some of these foods with that protein meal. If you're having animal protein, etc. I usually recommend having some juice and some whole fruit, waiting 10 to 15 minutes for that to kind of digest and settle down. Then having a protein source with some cooked veggies and then whatever fat you're going to have in your meal to basically manage the colon effectively so that you're able to get the adequate amount of protein that your body needs, the amino acids to maintain lean mass, etc., to minimize catabolism, to build new muscle. But then you also want to minimize the negative effect of these protein compounds hitting the colon, and that's where these different plant compounds come in, the tannins, the polyphenols. The fibers also have a beneficial effect as well. So yeah, and this is, I think, a better strategy as well than taking prebiotics and probiotics. The probiotics, if you're taking them orally, I mean, most people are taking them orally because the other route is kind of, a little uncomfortable. But if you're taking them orally, then you run the risk of them having them take up residence in the small intestine, which isn't ideal. You want them in the colon, in the small intestine. That's an absorptive environment. And this is a heuristic. There are some bacteria that take up residence there, but all in all, the goal would be to have a largely absorptive environment in the small intestine with minimal bacterial interference there, or microbial interference. And then in the colon, have a larger, you're going to have a bacterial population there. So the way to manage it is to have these foods that have these fibers and polyphenols and tannins, etc., that aren't going to irritate you. So ones that you tolerate, but at the same time are going to be able to modulate the microbiome in a beneficial way. So this is the kind of every single food that we eat or that we're going to be exposed to has some pros and cons. So it's about taking the pros and the cons and of the different foods, combining them, canceling out the cons, optimizing the pros, etc., and building into intelligent strategies from there. So that's it. That's all I have for you guys for today. Hope you enjoy this type of, I guess, content going over the different studies. If you have any questions, comments, or concerns, you can drop them below. If you want to subscribe from our content like that, please go ahead. If you like the video, please give it a thumbs up. And that should be it for today. Hope you guys are all well. See you in the next one.